Considering the emerging role of gut microbiome in various human diseases, several gut microbiome modulators like probiotics, prebiotics, diets, and drugs can help in ameliorating gut microbiome associated health ailments. However, no quick and affordable screening system are available to predict the effects of such gut microbiome modulators. But this system is simple enough to predict how particular interventions can impact gut microbiome that ultimately can impact the host health. This protocol is simple, affordable, and practical for laboratories investigating the gut microbiome modulators and their effects on microbiota and host health.
Maintenance of aseptic and anaerobic conditions is crucial for this experiment. Also, the fecal specimen should be used afresh immediately upon collection. Otherwise, if the experiment is planned later, the sample should be stored at -80 immediately upon collection, without any exposure to ambient temperature and air. Handling accuracy during aliquot prepping and pipetting is also important for maintaining accuracy and reproducibility.
It will take less than 12 hours in the daytime. We can leave the culture overnight if we need the samples in 24 hours, then followed by the short-chain fatty acids and microbiome determination.
This procedure will be demonstrated by two graduate students, Shokouh Ahmadi and Rabina Mainali from my lab.
To prepare fermentation media under a fume hood, in a reagent bottle on magnet mixture, mix solution A, solution B, trace mineral solution, water-soluble vitamins,